You are talking here about a topic that isn't brought up all that often, or at least I haven't been hearing it so frequently. N-RAS targeting patients with N-RAS mutated tumours. What is this all about, and what are the reasons that you've taken an interest in this and indeed done a study?
Yeah, the N-RAS mutation is a hot topic because it's 20% of patients who are carrying N-RAS mutations. So beside the BRAF mutations, which is found in 40% of all melanoma patients, it's the second most mutation, and we have targeted agents to N-RAS mutation. It's typically MEK inhibitors. And what I present here is an overview on the drugs which are under development for this disease at the moment. And there's 2 phase 3 randomized clinical trials to approve N-RAS inhibitors. And one is on ecancertv, a drug from Merck Serono, which is under development. This compares to DTIC. It's a trial which allows a crossover, which means if you are treated with DTIC, dacarbazine, standard chemotherapy, and you have a progressive disease, you can cross over to the MEK inhibitor. And the other one is called NEMO. It's a clinical trial which is testing another drug which was originally called MEK 162, and now it's binimetinib, a drug from Novartis which is tested against DTIC, but there's no crossover. And these 2 clinical trials are the focus for the N-RAS inhibition at the moment. But you know, my expectation is also that there is development of combinational treatments, and there's a CDK4/6 inhibitor underway to be combined with binimetinib, and the response rate was as high as 41% presented at the recent ECHO meeting.
So what might be the clinical messages emerging for doctors out of the whole concept of N-RAS targeting?
I think that's a very important question because, you know, for melanoma patients who are carrying BRAF mutations, you don't need to investigate the tumor specimen for N-RAS mutations. They are more or less exclusive. Which means if you are BRAF negative, you have a wild type, you know, you can be evaluated, and this enhances the chance to carry an N-RAS mutation from 20 to 40%. So it's of relevance for these patients, but the results at this point of time are not as good in the absence of phase 3 clinical trial results. From phase 2, the data are not as good as the data for BRAF inhibition. So the story is not the same.
So in clarifying a clinician's approach to an individual patient right now, how does all of this help at the moment?
So the drugs which are currently tested are not approved yet for melanoma, for N-RAS mutated melanoma. But certainly, I mean, this would be a good offer to include patients to a clinical trial, and if the drugs are approved, to use this drug. But there is huge competition, and the competition on the table is very clear. It's the PD-1 antibodies which work independently of any mutations, and so it's also a drug which could be considered for patients with N-RAS mutations.
But you're using MEK inhibitors, but this is not synonymous, though, with N-RAS mutation targeting, is it?
Yeah. You know, it's interesting because for binimetinib, the MEK inhibitor, you know, there was a clinical trial performed, and this clinical trial showed that there is effectiveness in patients with BRAF, and the rate was in a range of 25% response rate, but also with N-RAS, the same response rate in a range of 25%. So it was a randomized phase 2 trial treating patients with N-RAS and those with BRAF mutations. And this was the first sign that BRAF inhibitors can be used for both diseases.
So can you pull all of this together? If you're doing targeted therapy, if you're using targeted therapies, you look for the RAF mutation, you look for the N-RAS mutation, and you decide on a mix of different inhibitors. How do you go about it potentially in the future?
Yeah, the dilemma is that the only MEK inhibitor which is currently approved in Europe is a MEK inhibitor which is combined with BRAF inhibition. And this trametinib MEK inhibitor has never been tested in N-RAS mutated patients. So we simply have no data, and I wouldn't go ahead and using it in patients when I don't have data on it.
But some of the other agents might in the future give you that important data.
Absolutely. This is what we are expecting.